Please bear with me, as I am truly a computational scientist, and so I'll try to make sure I describe all the terms well as we walk through this, this movement, moving from Louis Pasteur methods to CSI and really getting into molecular approaches for diagnosing diabetic foot ulcers and bacterial infection in general. So our bodies are comprised of billions of microbes, both inside and out through our entire system. And in fact, these bacterial cells outnumber our human cells by more than 10 to 1. So we are truly more bacterial than we are human. Many of these bacteria, although we think of them as pathogens, are in fact commensal bacteria and are more helpful than harmful and really help with our digestion. They educate our immune system, and they provide a number of beneficial functions for us. The Human Microbiome Project is a new project that's been developed by the NIH, and really the goal of that project is to use these new sequencing technologies to sequence 250 humans across 20 different body sites and understand what makes up this human supraorganism. And what they began to uncover is that there is really a strong connection between microbes and our health and disease. And in particular, what's fascinating is that really there is quite a correspondence between microbes and our own individuality. So really our propensity for obesity, for Crohn's disease, can be determined not only by our genotype, but by also the microbes that are within us. And in addition to that, these new microbes or the good communities of microbes might actually provide us with new therapies or ways of treating disease as we know it today. So our skin, the skin microbiome, is made up of a number of organisms across the entire domain of life. So everything from bacteria to viruses to fungi to even the mites that live on and in us. And these different types of microorganisms play a really key role in our skin cells. And one of the things that they do in terms of the good bacteria is that they occupy a niche space on our skin that prevents pathogens from getting through. So they really compete with the different pathogenic organisms for those space and nutrients. And that provides a really important function in our bodies and our skin, fighting off disease and really preventing that pathogenic invasion. So one of the major problems with studying bacteria in particular is that most bacteria fail to culture. So what does this mean? So essentially, when you look at, you count the number of organisms under a microscope, it doesn't in fact match up with what you see in culture. So this is particularly relevant for anaerobes that are very slow growing, and so we may not actually be making the proper diagnosis based on what we see in culture. So these might not in fact be the most abundant organisms, but instead I would call them the lab weeds, really the ones that grow well given the type of media and circumstances within the microbiology labs that really help them to grow. So for example, Staph aureus here in this particular study, this recent study by Desmond et al. Of 970 different ulcers showed that in fact the majority of ulcers are Staph aureus, but are they really, or is this really just of course, it's a pathogen, but it is also an organism that grows very well in culture. So could we be biasing our results? So we've studied different organisms through genomics for many, many years. And what we do for that is we essentially culture the organism, and then we sequence that organism, and that creates a genome. And we've created a number of beautiful reference genomes this way. But perhaps even more exciting are the new technologies that are allowing us to sequence DNA directly from the environment. And in doing this, this is called metagenomics. And so the reason why we're able to do this massive amount of sequencing is really that the cost of sequencing has plummeted over the years, and the capacity for the sequencing machines has increased. So the reason for this is something that's called next-generation sequencing, which emerged in about 2005. And what you can see is there's just a really big change in terms of what we're able to sequence and at what cost, which means that in this, as we move to using this technology for clinical diagnosis, it becomes a potential reality. So there are two different flavors of metagenomic sequencing. One is using the 16S ribosomal gene, and what that gene is a gene that's conserved across all prokaryotes, but is not present in eukaryotes. And it's useful because you can actually compare that gene to a number of different organisms that exist, a number of different taxa that exist in reference databases, such as the ribosomal database. And this really gives you a count of who is in the sample and at what abundance. The other flavor of metagenomic sequencing is whole genome shotgun sequencing. And this is literally shotgunning everything, looking at everything, all the DNA within a sample. And when we do that, we actually get, we can see all the different organisms, and now we can also see their function. So we can start to understand whether or not these communities in these samples have methicillin-resistant genes or other types of antibiotic-resistant genes that could be important in their clinical diagnosis. So the ribosomal RNA gene really it makes use of the diversity of a particular type of gene. These are called hypervariable regions that are circled here, V1, V3, and V6, and that actually allows us to distinguish between the different types of bacterial taxa that are out there. In terms of metagenomic sequencing, I like to think of it as a puzzle where we have essentially taken all of the different organisms within a sample. We have many, many different fragments, and now genome scientists like myself have to come in and try to put the pieces of the puzzle back together. So what do we have? In fact, we have sequenced California, and the dominant organism happens to be the Golden Gate Bridge. And so we've put together the pieces, the fragment of these genomes, and we're actually able to see a pretty good picture of the Golden Gate, whereas the other sort of not so dominant organisms are less represented. So really the struggle with metagenomics is trying to put together and understand this fragmentary data in a way that allows us to really see all that function and understand where and what organism it originally came from. So it becomes a very difficult task. One of the things that is now emerging from the Human Microbiome Project using the 16S sequencing analysis is that the human, the skin sites across the human are actually different ecosystems. So what you see is that we have these different regions, for example, the dry sites across our body that have the most diversity. They're the rainforests of the bacteria across our bodies, whereas these sebaceous or oily regions that are less hospitable to bacteria are less diverse, and only a few bacteria can exist within those realms. And another thing that's emerging from the Human Microbiome Project in terms of skin is that these bacterial communities really vary between people. So here, what you see in this particular study is that there are 4 different individuals across 4 different areas of the body. And what we see is that although we see some broad patterns of similarity between those people, they do actually vary. And so what this means is that we as humans have different ecosystems of bacteria, and we may in fact need a sort of personalized medicine approach to understanding and diagnosing our personal bacterial communities across our bodies. So what is making up this ecosystem? So in fact, it's really a combination of many, many different pieces. It's the host physiology, the sex, the age, the site of the organism that we're looking at. It's the environment. It's understanding the host genotype and how it interplays with these microbes. It's also looking at the immune response. So not just what we have, but how our body is fighting it, and that delicate balance between tolerance and initiation of the immune response. And then also we have different types of underlying disease factors that may play a role in how our bodies respond to these different types of microbial communities. So when we think about microbes in terms of wound healing, traditionally we think about bioburden, or we think about microbial load and diversity and pathogenicity. But really, one of the things that's been emerging is that it's not just necessarily who's there and how much, but really the interaction of the community. And so one of the things that we can see from this mouse study that looked at differences between healthy mice and mice that were modeled for type 2 diabetes is that really we have different types of communities in these two different mouse models. The first, in the healthy mouse, is more diverse. So there are many, many different types of bacteria that exist in those communities, whereas the diabetic mouse actually has much less diversity and an increase in Staphylococcus. And one other interesting point is, as we look at the progression of this longitudinal analysis of the wound as it heals in the mouse, one of the things that we find is that day 14, when we get wound closure in our healthy mouse, we see this sort of uprising of the Lactosporidaceae. And then we see this, but in the diabetic mouse, we see an uprising of Klebsiella. So is that Lactosporidaceae having some sort of effect on the Staphylococcus where it's driving that down, and that's what actually is causing that mouse to have this delayed wound healing response? So there are a number of questions that we have as we start to really untangle this complex web of all the different types of microbes and how they not only interact with each other, but the functions that they have in the way that they communicate with their host, given different disease states. So really, when we think of wound healing, now we need to kind of expand our scope out a little bit more. So we need to think about the communities. We need to also think about the functional repertoire. So what types of genes are in these communities? How are they interacting with their host? How are they interacting with each other? And what's causing the differences in the abundance and type of bacteria that exist in these communities, whether they're diseased or healthy? And then lastly, we really need to think about the immune response. So the type of bacteria that are present really have, there's really a lot of crosstalk between the host and the bacteria that are present in a sample. And we need to consider that when we look at all of these things together. So in terms of diabetic foot ulcers, one of the most important things in studying diabetic foot ulcers is making sure that we have clinically homogeneous populations to look at, that we're actually defining and using you here in this community to help us define what those phenotypes are, to really understand what differentiates communities, to really try to dive into those clinical factors that might be driving these differences among communities. This study is really one of the only studies that I know of that does this, where they essentially look at neuropathic diabetic foot ulcers in non-infected wounds, and they associate them with clinical factors. And what they find is essentially that diabetic foot ulcer depth is associated with increased diversity, increased anaerobes, but then a decrease in Staphylococcus. And then the diabetic duration of the foot ulcer is associated with increased richness and diversity, but then also an increase in the proteobacteria. And then lastly, like our mouse model for the diabetic type 2 diabetes mouse, what we see is that given poor glycemic control, we also have this positive correlation with Staphylococcus and with Streptococcus. And so these types of things are starting to help us understand the role of these communities and how they affect disease. So with that, we are really trying to literally shoot for the moon with this. But there are a number of different challenges that we face as we go along. And some of the things are, are we really able to use these new technologies in the clinic? Are we really able to drive down the costs for care and sequencing? And then also, are these new methods, are they actually optimizing the antibiotic therapies? Are we really able to do this sort of personalized medicine approach to making sure people get the right type of antibiotics or don't get antibiotics at all, given an uninfected case? And then also, we really need to untangle not just the bacteria, but really understand the relationship between bacteria, the viruses that infect them, the fungi in the sample, and the entire microbial population across all of the samples. And then understanding that functional repertoire, really making sure that we understand the functional capacity of these different communities and how the interplay between the host happens. And then, are we really able to use all of this wonderful, beautiful data to predict outcomes? And are they representative of these really the live DNA or live samples? So one of the issues with doing DNA sequencing is that you sequence everything, live or dead. So what we have to understand is, can we use appropriate sample prep methods that really let us get to looking at what the live organisms are in the sample? And with that, I'd like to do some acknowledgements for the Arizona Genomics Corps, who I'm currently working with to really push forward these new methods, and then also the SALSA Institute, and in particular, Dr. David Armstrong and Bijan Najafi. So thank you very much.